In last week's lab demonstration, Nathan extracted DNA from an environmental sample. This week, he's going to perform the next critical step in any molecular microbiological approach, polymerase chain reaction, or PCR. Using PCR, we can amplify DNA by several orders of magnitude, making it suitable for use in a variety of downstream analyses.
The first step in any PCR is to add all of our chemical reagents to our reaction. In any PCR, we need to make sure that in addition to the DNA samples we wish to amplify, we include both positive and negative control samples. A positive control is usually a known amount of high-quality DNA template, often acquired from a bacterial culture, and is used to ensure that the PCR conditions are suitable for a successful amplification. The negative control is a buffer known to lack the DNA target and is used to ensure that false amplification does not occur due to the contamination of reagents. To each of the tubes containing DNA template, we need to add several reagents for the PCR to work. These include 4 microliters of the forward and reverse primers. Today we are using primers that target a specific region of the bacterial 16S RNA gene, and 25 microliters of Taq master mix, which contains the thermostable Taq polymerase, which is needed to copy the DNA, along with a mix of the 4 deoxyribonucleotides, or DNTPs, which provide the fuel for the amplification process. Within each tube, we will add 11 microliters of millicube water, 10 microliters of DNA template, 4 microliters of primer mix, containing 2 microliters of each primer in 10 millimolar concentration, and 25 microliters of Taq master mix. Once you've added your PCR reaction mix and your DNA to your tubes, we'll then vortex them and centrifuge to ensure that the entire mix is at the bottom of the tubes, because that is where the reaction will be occurring in the PCR machine. We'll then place the tubes in the PCR machine and program the PCR. For this PCR reaction, the following cycling conditions will be used. An initiation step for 120 seconds at 95 degrees Celsius, and then we'll have a denaturing step for 30 seconds at 95 degrees Celsius, followed by an annealing step for 30 seconds at 50 degrees Celsius, followed by an extension step for 90 seconds at 72 degrees Celsius. These 3 steps will be repeated for 19 cycles. Following these 19 cycles, we'll then have a final extension for 120 seconds at 72 degrees Celsius. After about 2 hours, the PCR cycle is complete and our DNA should have amplified. To check that the PCR has worked, we have to perform electrophoresis. The first step in this is making the gel. To make the gel, we'll weigh up 0.5 grams of agarose, and then we'll add this to 50 ml of our TAE buffer. We'll then microwave the solution in short 10-second bursts, making sure that the solution doesn't boil. Once the agarose is all dissolved, we'll then add a nucleic acid dye called Gel Red. This will bind to our product and help us to identify it after electrophoresis. We'll now add 2 microliters of Gel Red . Next, we'll set up the gel for electrophoresis. This includes adding the gel chamber to the rubber stoppers, tightening the stoppers, and then pouring the gel. We then add these teeth, and these teeth will help to form the lanes in which our gel will run. Once the gel is set, we'll then need to add it to the electrophoresis chamber. First of all, we'll put our TAE into the electrophoresis chamber, pouring it up to the max line. Once we've done this, we'll then remove our set gel from the clamp. We'll also remove the teeth so that we can pipette our samples into those wells. When placing the gel into the electrophoresis chamber, we want to ensure that the wells that we've made with those teeth are facing the black end of the electrophoresis chamber, because our sample is going to run from black to red. Next, we want to centrifuge down our samples to ensure that no sample remains on the side of the tube. We'll then take our samples from the centrifuge and then begin to load the gel. The first thing we want to load into our gel is our ladder. We put a ladder into our gel as a known reference so that we can view the gel and know how big our fragment size is. We add the ladder to the first lane of our gel, and then we discard our tip. We'll then add our sample, our positive control, and our negative control. Our sample will go in the next well. And our controls will go in the wells after that. Once we've loaded our samples into the gel, we'll then need to secure the lid on top of the electrophoresis tank. To do this, we'll need to make sure that the electrophoresis equipment is turned off so that we don't electrocute ourselves. We'll add the black end to the negative and the red end to the positive of the electrophoresis chamber. We'll then ensure that the lid is secured tightly. Switch on the electrophoresis equipment and set the settings to 100 volts and run it for 30 minutes. You can check whether the gel electrophoresis has been set up properly by looking for upward flow of bubbles from the cathode. This is the black end of the gel electrophoresis chamber. We now let the gel run for 30 minutes, which involves the DNA migrating across the gel. Once the electrophoresis is complete, we'll then need to switch off the electrophoresis kit and remove our gel from the chamber. We want to be careful that the gel doesn't slide out, placing it on the tray ready to transport over to the UV. We then use the UV illuminator so that we can view the band on our gel. If a band indicative of the correct DNA fragment size is visible on the gel, it means the PCR has been successful and the DNA has been amplified. The amplified DNA is now ready for use for downstream processing and analysis, such as DNA sequencing.